We work on something called melatonin. Here it's called phytomelatonin, but it was really discovered in animals. It's a very well-known molecule in animals, which was discovered in 1958 by a dermatologist. And in 1995, we discovered the same molecule in plants. You probably know melatonin because of its ability to adjust your circadian rhythms. It's often used to overcome jet lag as a sleep-promoting agent, and it is also a very potent antioxidant. And some of what we're going to talk about today relates to its antioxidant activity in plants. Of course, melatonin is also phylogenetically a very, very ancient molecule. We estimate it to be around 3.2 billion with a B years old, by virtue of the fact that it's found in cyanobacteria, some of the most primitive organisms that evolved at this time. The one that we looked at in particular is photosynthetic, and throughout evolution, including in you, melatonin is produced. And it's produced in a very unique manner in you. But we're not going to discuss you. We're going to talk primarily about its action in plants. But it exists in every species that has been studied. Every plant species, insects, reptiles, amphibians, fish, in unicellular organisms, in protists, and also in bacteria. Of course, since bacteria produce it, and bacteria are the precursor of both chloroplasts and mitochondria according to the endosymbiotic theory of the origin of these organelles, when cyanobacteria were initially ingested by primitive eukaryotes, they did it for the purpose of nutrition. They were eating the bacteria, in other words. But eventually, they evolved into the chloroplasts, which now, of course, have been retained. And since the cyanobacteria produce melatonin, we think melatonin is produced in chloroplasts. And we now have evidence for that as well. Additionally, mitochondria evolved by similar means from a bacterium also, which is also known to produce melatonin, and as a consequence, we think the mitochondria, the result of engulfing these, are also sites of production of melatonin, and every cell in your body has mitochondria. Since animals only have mitochondria and plants have both chloroplast and mitochondria, plants generally have higher concentrations of melatonin than do animals, including you. The synthesis of melatonin in animals is very well described. Of course, it's a tryptophan derivative, and in plants, or at least in chloroplasts, there's some evidence as to how it is produced as well. There are differences. Certainly in animals, the rate-limiting enzyme N-acetyltransferase. In plants, it seems to be tryptophan decarboxylase. But you notice these two enzymes are reversed in plants and animals. But this seems to be primarily the rate-limiting enzyme. There are some other activities in plant melatonin synthesis that I'm not going to discuss. For your purposes, this is the pathway we're going to be concerned with. Melatonin is a remarkably effective antioxidant, much better than vitamin E, than vitamin C, than glutathione, or any other one you can measure. And this has been very well documented, a discovery, again, that we made in 1993 in conjunction with Tan, who's sitting right here, and it's been confirmed thousands of times in the intervening year. Melatonin scavenges free radicals, and when it does, it generates something called cyclic 3-hydroxymelatonin. It happens that this molecule is also a very good scavenger. It generates N-acetyl N-formyl 5-methoxykynuramine, and it is a scavenger to produce N-acetyl 5-methoxykynuramine, and it is a scavenger. This is what makes melatonin so effective in reducing oxidative damage. There's a whole cascade of molecules that are all function as very efficient free radical scavengers. Vitamin C scavenges one radical. Vitamin E scavenges one radical. Melatonin, via this cascade, referred to as melatonin's antioxidant cascade, we calculate, may scavenge up to 10 free radicals. Additionally, it stimulates antioxidative enzymes, which I'm not going to discuss extensively. But this occurs in plants, in animals, and obviously in you. Melatonin in humans has been linked to aging, to prevention of neurodegenerative disease such as Alzheimer's and Parkinsonism, and in plants it also has antioxidative functions. Again, in this study that we published about 10 years ago, we looked at the water hyacinth to determine whether in fact melatonin is converted to the metabolite, for example, AFMK, in the plant as in animals. Throughout a 24-hour period, this is the period of darkness here, throughout the day, melatonin levels increase. And this increase is a consequence of the intensity of the light. We know if we grow these plants indoors under lower light intensity, they produce less melatonin. This is probably a stress to the plants, and as a consequence, they generate more free radicals, and melatonin is upregulated to scavenge those radicals. Shortly after the melatonin peak is in fact the peak of AFMK, because AFMK is the product that is generated, one of the products that is generated when melatonin scavenges radicals. We use this as evidence that in plants, as in animals, melatonin is a good hydroxyl radical scavenger. This is demonstrated now in many, many, many studies to be the case. In the case of the sweet cherry, this was work done by Jin Kong in Hong Kong in conjunction with us, and she looked at the melatonin development or the melatonin changes during growth development in sweet cherries. The blue line here represents the melatonin content of cherries as they go through this maturational process. There are three phases: stage 1, stage 2, and stage 3. Stage 2 is the point of lignification of the pit, when the lignin is laid down in the pit, and during that time, early in that phase, free radical generation is very high, and it wanes as the pit becomes lignified. As a consequence of that increased oxidative damage or increased oxidative production, you get a large increase in melatonin to protect the other tissues in the cherry from oxidative damage. Throughout the remainder of development, the production of melatonin is low. Conversely, the malondialdehyde, which is lipid peroxidation product, is generally inversely related, and you would predict this. Melatonin, of course, prevents lipid peroxidation by virtue of its ability to scavenge free radicals. The rate-limiting enzyme in melatonin production, namely tryptophan decarboxylase, is also high when melatonin is high and wanes during the remainder of the developmental period. Again, suggesting that, in fact, melatonin in this plant, as in others, is a free radical scavenger. Melatonin also has auxin-like effects in plants. Of course, both of these molecules, indole 3-acetic acid and melatonin, are derived from tryptophan. They are structurally very similar. Both are phytohormones. Both can move around in the plant. Auxins, of course, are not produced in every cell. They have to be translocated, and we know melatonin is also moves around in plants. It can be absorbed from the soil, for example, and get it in the plant that way. And both of them are upregulated under certain conditions. If a plant is wounded, oxygen levels are upregulated, and of course, in the event of oxidative stress, melatonin is upregulated. So melatonin influences the plant growth via oxidant-like actions, whether it's a fact that true oxidant remains unproven. That is represented here. This is a study by a work of Kang, also in China, where he looked at the effect of incubating or transgenic plants containing AANAT, the enzyme that converts serotonin to N-acetylserotonin, the precursor melatonin. This would increase melatonin production, and as a consequence, when those seeds are germinated in a cold environment for 8 days, they significantly grow better. There's additional chlorophyll being produced, a 5-fold increase in chlorophyll generation, and likewise, chlorophyll 2-fold increase, the melatonin content increases 5-fold. So melatonin improves, we now know, primary, lateral, and adventitial root growth in these plants. If you improve growth of the roots, you would expect to improve growth of the plants as well. Everything suggests that melatonin improves growth of many different plant species. Conversely, a melatonin deficiency has negative effects. In this case, the plants were transfected with indole 2,3-dioxygenase, which metabolizes tryptophan to another root and depletes the serotonin. This is a normal tomato leaf complex. And of course, after a melatonin deficiency due to a expression of indole acetic acid, or indole deoxygenase, you can see the abnormal leaf development that occurs. Multiple leaflets and much flatter and less serrated leaves in most cases. Occasionally, however, you get a normal leaf. So both exaggerated and deficient melatonin impacts the growth of plants. Melatonin can be upregulated by chemical toxins. I suspect arsenic is one example, although arsenic has not been tested. In this case, Arnu in Murcia, Spain, tested hydrogen peroxide, calcium chloride, and zinc sulfate, and in every case, within a very short interval, melatonin was dramatically upregulated in the lupin bean. In other words, the melatonin content increased proportionally to the severity of the stress of these molecules. Salty soil, of course, is becoming a very major issue around the world. During drought, the soil becomes much more salty, and of course, the deeper you drill your wells and irrigate your soil, the more salt that is retained. So this is particularly important. And melatonin, we know, protects against these types of stresses, biotic stresses. Melatonin clearly enhances plant tolerance to biotic stress, including biotic stress resulting from a variety of different means. In this case, melatonin prevents leaf yellowing after 90 days of drought. This is an apple plant. These are control leaves from plants that were irrigated adequately. This is irrigation with melatonin. There's no difference in those, but drought for 90 days causes a very dramatic change in the leaf. And of course, if they have been irrigated with melatonin, they are, of course, prevented. The melatonin was given before the drought began. So melatonin reduced glutathione loss, reduced ascorbic acid reduction, reduced leaf senescence by the SAG12 gene, and reduced chlorophyll degradation as represented by that gene as well. In other words, again, melatonin protected against this type of biotic stress. Likewise in Bermuda grass. Bermuda grass is commonly used throughout the world for athletic fields and so forth. Under controlled conditions, the addition of melatonin does not change. These plants are growing maximally with adequate irrigation, and there's no change in fact if you add melatonin. Conversely, if you expose them to sodium chloride for 25 days, up to 300 millimolar, there's a significant reduction, but if they have been treated with melatonin, particularly these higher concentrations, you can see there's some preservation of activity. Likewise, during drought, just as with the apple please, you can retain some activity by treating with melatonin, and even freezing. Now, that wasn't freezing for 25 days. It was 21 days of plus 10 degrees, then followed by 10 hours of freezing temperature, and then back to 10 degrees. And again, when melatonin was added, they recovered much better. This is the work of Haitao Shi from Wuhan in China as well, in conjunction with our laboratory. They've done a lot of other work. The leader of that group is Dr. Chan. He's also at the Wuhan Medical Center, and he has looked at a variety of things that melatonin protects against. Salt, we've already mentioned, cold, drought, and heat, all are protected with melatonin. The implication is that if plants are enriched with melatonin, they could be grown in much harsher environments in general, regardless of what the insult may be. And in fact, he has done transcriptomic and metabolomic analyses of 3,933 genes, most of which are upregulated by melatonin, and they refer to sugar metabolism, tricarboxylic acid cycle, and sugar alcohol metabolism and carbohydrate production. So melatonin, we now know, has a whole variety of beneficial effects in plants, and importantly, it's upregulated when it's needed most. There's also evidence that it protects against biotic stress. There are only 2 studies in this area. There are many studies in the abiotic stress. Many plants are known to be protected by endogenously produced upregulated melatonin. In this case, the biotic stress was in fact a fungal infection. Mycelium spores were smeared onto the leaves of these apple plants, and the apple plants were irrigated with or without melatonin in these concentrations. This is without melatonin. This is, of course, with the lowest concentration. And as you can see, the higher the concentration, the less was the fungal infection. So melatonin protects against not only abiotic, but biotic stressors as well. This is the work of Yin, also in China. And melatonin delays senescence of detached apple leaves. This is the work of Ma Wang as the first author, but it's Ma at Yangling, China. And they remove apple leaves, put them in the dark for 12 days with and without melatonin. And of course, you reduce the loss of chlorophyll by suppressing the gene that degrades chlorophyll most dramatically. You suppress the senescence-associated gene 12. I told you that on the previous slide. So they do not senesce as quickly. You maintain ascorbic acid and you maintain glutathione. Everything melatonin protects against relates, for the most part, to the free radical accumulated damage and senescence changes. Here's a summary of the abiotic and biotic stressors induce melatonin synthesis. Namely, they are the ones that have been tested: high salinity, drought, metal toxicity, particularly cadmium and copper have been tested, but I suspect the similar would apply to arsenic or any other metal. Extreme temperatures, either cold temperatures or hot temperatures, melatonin protects against. Pesticides, excess water, poor soil quality, and biotic stressors. The one I showed you is a fungus, and there's one, apple blotch disease, which is and against a fungus. So melatonin increases tolerance generally of plants to abiotic and biotic stress. Via this presumed pathway, melatonin induces antioxidative enzymes and upregulates melatonin production as well. And here are the consequences at the molecular level which melatonin preserves. I want to show you one other study that's very important, namely that melatonin also increases crop productivity. In this case, we used soybeans, and soybeans are harvested at the rate of 370,000 tons of soybeans are harvested every year. And as you will see, melatonin, seeds grown from melatonin, plants grown from melatonin seeds will increase crop production by about 10 to 15%. These are some of the measures. Even germination is improved. This is germination of seeds not coated with any melatonin, but given the seed coating reagent, which is typical of soybeans. All soybeans are incubated in a seed coating reagent. They germinate at the rate of about 60 to 65%. After melatonin addition to the reagent, that germination is significantly increased. That alone would be very important in terms of total crop production. Of course, melatonin improved germination and the growth of the soybeans. You can see the difference in the soybeans here, and the number of bifolds and the number of unifolds as well were increased, as well as the top third trifoliate was enlarged. So the plants were generally healthier, grew faster, and better. At 3 months, we looked at, in fact, the crop production. Here again are the products of being incubated with no melatonin, 50 or 100 micromolar. These plants are clearly larger, and here is the production of these plants. In other words, there was an increased number of pods, an increased number of seeds per pod, but the size of the seeds did not change. In other words, if you measured 100 seeds, they had the same weight, but there were more of them and there were more pods. And similar results have been obtained with cucumber and corn by Margosia Pozmik in Poland. So evidence is that melatonin increases production of plants as well. And as I say, if that would actually apply to soybeans in the natural environment, it would mean 37,000 tons of additional soybeans would be produced just by treating the seeds with melatonin. Melatonin is very inexpensive and non-toxic to the soil or to the animals or plants. Melatonin is also taken up from the soil. If you add it to the soil, it's taken up by the plants. Drought stress for 7 days increases, because it's a stress, increases the melatonin content of the seeds, but also exposure of the seeds to melatonin, 1, 5, or 10 micromolar, greatly increases. Melatonin is readily absorbed by seeds. Many people have shown this as well. And when they have more melatonin, they germinate better, they grow larger, and produce more product. This is a summary by Marino Arnu and Josefa Hernandez Ruiz in Murcia, and this illustrates the many beneficial effects of melatonin in plants. You have to understand, this data has accumulated very rapidly, most within the last 5 to 7 years. Melatonin was only discovered in plants in 1995. The bottom line is genetically engineering of melatonin synthesis may be a primary target to improve plant tolerance to biotic and abiotic stress, and also to improve crop production without any additional land included for production. Conclusions. First of all, I want to point out that my email and Dr. Tan's email address is here. You're welcome to contact us about this information at any time. We obviously have many publications on the subject if you're interested. Melatonin is present throughout the plant and animal kingdoms. Melatonin is a universal antioxidant in plants and animals, and it's a very potent antioxidant as well. Melatonin preserves chlorophyll synthesis, which allows the plant to survive better. Melatonin improves plant tolerance to both abiotic and biotic stressors. I showed you some examples. Of course, there are many more. Melatonin generally improves what we call cellular infrastructure. It improves the stability of cells, not only in plants, but in you as well, in terms of survival and coping with toxic environments. So melatonin improves plant growth, we've shown that, and this is probably more manifested When the system is challenged, the effects of melatonin are more apparent in both animals and plants when you challenge the system, and then melatonin can do its job. Melatonin improves crop yield, and melatonin aids plants to survive and to thrive, as implied in the title of what I said. Melatonin is remarkably efficient at protecting you and protecting plants from all kinds of stressors. And it can be induced, certainly in plants, and also evidence that it can be induced in humans and other animals as well. Thank you very much.
Is there anyone applied for plant tissue culture?
Please say again.
Is anyone applied this compound to the plant tissue culture?
Has it been tested in culture?
Yeah.
Many times. Melatonin in terms of cultured plants.
The tissue culture.
Yes, of course. Many, many studies.
What kind of effect?
All the same as represented here. Melatonin is always positive. Not always positive. I'll give you an example. I don't mean to talk about something else. But melatonin prevents death of normal cells in humans, but it kills cancer cells. In other words, its actions are context-specific in many cases. This is really remarkable for a molecule to enhance the strength of normal cells and, on the other hand, kill cancer cells. And I suspect in plants, it probably has similar context-specific actions. Please.
Basically, I have two questions, if I am allowed. The first question is that, do you have any idea how many genes are involved in synthesis of melatonin? And the second question is, if these genes are involved, what are the inducer of these genes? Do you have any idea? Because I'm thinking about the engineering of genetic engineering.
Yeah, I know.
Yes.
Well, genetically engineered are ready to improve their plant production. The microtom tomato and one variety of rice has been gene manipulated by transgenic gene manipulation to upregulate melatonin production. How many genes are actually involved in the pathway of melatonin production? I didn't even show you the complete pathway. I showed you the basic pathway. So I don't know how much genetic engineering would be required to enhance melatonin production in plants. But I think, knowing what I've seen of you people, it would be perfectly feasible to do. But I'm not a plant genetic. Well, there's 4 enzymes, but there may be more genes. And your second question was? How is it induced? We think it's, again, we're not plant physiologists, but when people talk about inducing abiotic stress, inducing something in plants, they usually involve a receptor. I don't know what that stress receptor is. Every paper I look at has a stress receptor in the cell membrane. I personally do not know what that receptor is. You may do it. Calcium is released internally. It activates a number of genes which stimulate antioxidant enzymes and melatonin production. The whole progression there we don't understand at this point. But we do know that melatonin and its antioxidant enzymes are clearly upregulated under abiotic and biotic stress conditions. And the genes, the senescence accelerated gene is downregulated, and the gene or the gene which leads to the degradation of chlorophyll is also downregulated. So we have some idea of the mechanisms involved, but not all.
That the melatonin helps as a sleep aid.
As a sleep aid.
And at the same time is antioxidant.
Yeah.
So what would be the required dose per person, not in the daytime, in the evening, 5 milligram or 10 milligram?
He's asking about melatonin as a sleep aid. You understand the original function of melatonin was an antioxidant. In the cyanobacteria, they were photosynthetic, they were generating a lot of oxygen of course, which photosynthesis does, and they needed protection. So melatonin evolved as an antioxidant, and we have some idea why the rhythm evolved also. And it was finally usurped by other animals to regulate circadian rhythms and sleep. It's not a soporific like the drugs that promote sleep. The way melatonin promotes sleep is it opens a normal sleep gate. So you have to take it at the right circadian time. That's usually the evening, but it's only affecting about 65% of the people if you take it to the wrong circadian phase. But it does promote sleep. It promotes natural sleep. And it does that in diurnally active animals and in nocturnally active animals, even though every species, whether they're active at night or during the day, melatonin is high at night. In animals, melatonin is a chemical expression of darkness. How much does it take? 250 micrograms, 5 milligrams. Some of us take much more. Not necessarily for sleep, but especially as an antioxidant. You're the boss. Can we afford more questions? Okay.
Let's have a question.
Just a quick question. I wonder whether there is any non-negative effect from melatonin.
Is there any negative effect from melatonin? Melatonin, as described by the pharmaceutical industry, is the least toxic molecule they have ever tested. For example, in animals, you cannot kill an animal. You cannot make an animal sick by giving them too much melatonin. On the other hand, in plants, very high concentrations in the soil have been shown to stunt somewhat the growth of plants. Why that occurs, we do not know. But generally, it's a very, very safe molecule. And you have to understand, because there are a lot of microorganisms in the soil, there's a lot of melatonin always in the soil. All these bacteria, worms, whatever's in the soil, are producing melatonin.
Excellent lecture. I could learn a lot with you. I work with induced resistance in plant. And we inoculate one fungus which induce resistance in the plant against a second fungus, which is a phytopathogen in the plant. But I've never stopped to look for melatonin. But anyway, melatonin is in high concentration, a low concentration on how the people look for melatonin. They use LCMS. You show something.
There's many, of course you can do rate immunoassay, enzyme-linked assay, but they are obviously less expensive. Liquid chromatography with tandem mass spectrometry methods, any of those methods are better than the conventional ones. But many, and the amounts of melatonin can vary widely in different plants. We anticipate that plants that grow in a more northern environment and under more stress from temperature or cold will have more melatonin. But there's wide variations. And I didn't tell you the whole story about melatonin. There's some other molecules that are produced in plants that are derivatives of melatonin. All we know at this point that is very protective of plants against a lot of types of stress.